Hi, welcome to the 34th episode of Startup Strides. Today, actually, it's one of the first episodes where I have two guests at the same time. I have Christian Ponce, who is the CEO and co-founder of Tetsuwan Scientific, and Théo Schäfer, who's the CTO and co-founder of Tetsuwan Scientific. Um, really excited to have you guys on the podcast today. Genuinely looking forward to this conversation, and welcome.
Thank you. Thanks for having us on.
Yeah, thank you, JV.
Of
Very
course.
happy to be here.
Of course, same to I've read up on Tetsuwan myself a bit, obviously from the website, from some of my personal research. But for our audience, I'd like to hear it from you personally because I know there's an interesting backstory to how you guys got it started. So, how did Tetsuwan Scientific come to be?
Yeah, absolutely. Christian and I met in New York back at Halloween night 2023. We met in a startup incubator called Entrepreneur First. They're running this program. And we met through that program. Christian had a very interesting background in biology, bioengineering, and I didn't know anything at the time. The purpose of the program is to meet people, compare your skill sets and try and see if there can be an interesting overlap and how you can potentially work together. And yeah, when I met Christian, I thought he was amazing person, and we started comparing our backgrounds. Told me about bioengineering, and he was so passionate about the projects he was working on, the minimization project. He was telling me, I didn't know anything about biology. So he tried to explain very simple word. It was synthetic biology. It's about taking organism, engineering them genetically so that they can produce new functions or new molecules. It's super, super interesting. And when you're modifying an algorithm, there is some genes that you want to remove because they might interact with what the function you want to be. And so I was we were working in this minimization project, removing all the genes, and it was super exciting, but the pain of the work was unbelievable. I had to do everything by hand. And at the time I was coming from NASA and MIT. I did robotics projects there. I was working on autonomous underwater vehicles, autonomous systems in general. And we were sending robots to other planets to do automated work, right? And the lab is a super controlled environment. And I was wait, you don't have robots there? I was no, man, I was doing everything by hand. I was we need to fix that. And we started exploring this field.
Honestly, kudos to you as coming together and just going after such a very advanced technology too, which we're gonna delve into later on throughout the episode. But I very much resonate with what you said about Christian about research. I was a biochemistry major in college in the US actually, did research here and there, and then I ended up actually, I don't know if you guys see the Yale thing, but I did the research at Yale for a year after graduation. Kind of hated it. Realized wet lab research is not really for me. The pipetting, the qPCRs, the cell cultures, it's just, I feel like there's a bit of a disconnect between what we imagine science to be and what science actually is, right? When you see the news about the breakthroughs and inventions and the clue research, behind it is years and years of tedious, mundane, kind of slow work that just builds up on each other. And also kind of taking that into consideration and also saying is that I wouldn't make a good scientist in general. I realize I'm not going to pursue a PhD. But what are you guys doing is this can essentially fundamentally change how we do science in general, right? But, I'm curious, what exactly are you trying to solve and how are you approaching it?
I think our main frustration is that science today is brain power inefficient. It's like you said, there's a disconnect between what we imagine science and research is when we're growing up, and then what it is once we go into it. And this disconnect is very disheartening. Science should be and will be about discovery, not about repetitive labor. And unfortunately, it's not just that this repetitive labor makes it so that people like you and I are no longer super enthusiastic about biochemistry or bioengineering, but it also introduces a lot of sources of error. Reproducibility is a major problem in life sciences. So the way that we use scientific labor now isn't only brain power inefficient, it's resource inefficient. We're moving slow, expensive, and executing experiments that aren't super reproducible. So this is our main frustration. Human scientists should be, talking about literature. They should be reading more literature. They should be in lab meetings. They shouldn't be at the bench. there should be automated solutions at the bench to allow human scientists to focus on what we associate with being the work of a PI today, which is directing labor to discover.
Gotcha. And not long ago, this concept of AI-powered science. By the way, if I look to the side, it's because I'm looking at the questions on my iPad, so I'm fully attentive on the conversation. This idea of AI-powered lab scientists before wouldn't have been it would be very much impossible. And I feel like because most experiments requires this constant parametric changes, and fully automating that is a quite bit of a challenge. But in the recent years, was this rapid advance in AI, the natural language processing, LLMs, the entire field of science, biology, everything is really changing. So I'm curious, I'm just more curious about this technological foundational change, right? What makes this technology the key paradigm shift for lab automation?
Yeah, absolutely. Just like you described, trying to automate science and especially research and discovery in science is extremely complicated because of all these variations. You're never running the same protocol twice, right? Science is always going to tweak the protocol, change some parameters, change the volume, change the temperature of the reaction. There's always changes. And you can try to build a rule-based system to tackle this, but it'll never be completely covering all the edge cases. It's just impossible in terms of complexity to cover all aspects of what a protocol can look like. So rules-based system approaches have been tried and have failed. The beauty of the new systems like LLMs, natural language processing, is that it allows you to create rules as protocol changes. You can have a much more flexible system. It also enables the interface between the scientist and the automation tool to be much more simple. You can remove all needs for programming or even need for lab automation engineers. The systems like natural language protocol, they're context aware, right? When the experiment is mentioned, the LM can base answers on its knowledge and the data set it was trained on, and basically make decisions based on this context, based on this natural language feedback with the scientist. Ask question, oh, you gave me this protocol, but based on my knowledge, I know that RNA extraction, for example, would involve this step, and I see you didn't mention this step. Did you forget that step in the protocol? And the scientist would be oh yeah, sorry, these things happen. You would be very surprised. It's very difficult to completely list all the steps and exact details of a protocol. So those natural language processing tools help the translation between the scientist and the automation tools.
And a lot of what saying may come as a surprise to some of your listeners. It's hard to list all of the steps of a protocol. When we think about the protocol that we would read at the bench, our Zymo protocol or our QIAGEN protocol, whatever protocol we're reading from the kit, that may be shocking. It's no, it details it pretty well. But once you try to convert that to an automation script, there's a whole bunch of complexity, like Théo says, that we don't really capture. One of these is liquid class characterization. So there's a lot of things that we attribute to muscle memory and to intuition in this conversion of high-level human-readable instructions to low-level, exact, non-ambiguous instructions. And one of those is how we deal with liquids. When I'm at the bench and I pipette glycerol, I never think, oh, the polarity of glycerol is this. The viscosity of glycerol is this. Therefore, I should pipette it with this dispense speed and aspiration speed and blowout volume. We never consider that. But all this complexity arises when you try to translate implicit to explicit. And that is the primary thing that we're really interested in here. Our terminal interest is in creating autonomous systems for scientific discovery, but a very, very instrumental goal to get there is being able to interface human-readable protocols with automation platforms. Right now, as Théo mentions, an automation engineer does that. They stand in between human-readable, how we operate, and how the machines operate. But this translation is a huge bottleneck. And unfortunately, the same way we need these automation engineers to interface with automation tools, models need automation engineers to interface with these tools. So unless we automate the automation engineer, automate this translation from implicit to explicit, from human readable to automation compatible, we can't give models physical agency to control work cells or other lab automation ecosystems.
Gotcha. This is really interesting, as sometimes beyond my comprehension levels. Um, so you guys touched upon on what you're trying to solve, and you explained the underlying technology as well. But I'm curious, just purely from like what Tetsuwan is doing in technology wise. You don't have to disclose any confidential information, obviously. Protect your IP. But so what? How does Tetsuwan come into a picture? Like what's Tetsuwan doing with this technology for the scientists?
Of course, our goal currently is the automation of this automation engineer, this middleman that translates what the human is saying, the bench scientist, the process development person without any lab automation experience, into something that runs on a lab automation ecosystem. Today, that is the bottleneck to using lab automation, this translation, this support. And because of this, how expensive this translation is, how expensive this support is, we only use lab automation for pretty high throughput stuff. What we're interested in here is in automating this process of translation between the bench scientist or process development and the lab automation ecosystem. This is a translation from implicit to explicit. And this might not make a lot of sense. So I'll give you a better example. We can think of a cooking recipe that might be on the back of a box of batter or something, right? Maybe it's a cooking recipe for pancakes, right? It doesn't tell us exactly what to do at every single step. It gives a few high-level instructions. It says you need eggs, you need flour, you need butter, you need to oil the pan, right? Or maybe it doesn't even say that, but it doesn't tell you what type of pan to use. Maybe it doesn't tell you what temperature to heat the pancakes at. It doesn't tell you when to flip them. It says cook till golden brown, right? But we're able to take this high-level set of instructions and convert them into exact motor movements. If we tried to hand these instructions that would be on the back of a cooking box, this recipe, to a compiler, the compiler, it wouldn't work. The compiler requires explicit, non-ambiguous instructions. We call these sets of instructions programming languages, right? So how do we convert this recipe, this cooking recipe, into exact motor movements? We call that intuition. We call that common sense. We're able to read that, extrapolate the exact things we have to do because of our common sense. The same issue exists at the bench. When we read our version of cooking recipes, our protocols, we use intuition to translate them from a high-level set of instructions into a low-level set of instructions that we can go and execute. But this translation that intuition makes possible is really, really hard to do manually. This is what we're trying to automate.
Gotcha. So essentially, what you're saying is your AI lab assistants can interact with human researchers that don't really have any experience in programming. Is that true?
Yeah, absolutely. But this is not the biggest differentiator here. Other companies have already, been advocating no-code interfaces. Like a lot of companies have built like GUIs and systems where the scientists can interact with their robot without having the need to code. They'll have a drop-down menu and they'll select like the labware and put it like wherever on the deck and like select the pipette and select whatever parameters. Actually, the difficulty here is not only like getting rid of the code, it's actually like all this knowledge, like what I just said, select whatever pipette parameter or place your labware wherever on the deck. Like those things, they're still like the charge of the scientist. And that's like really bad because that's like a lot of automation knowledge required to set up these kind of things. Like you need to think about so many parameters in order to set up your experiment like properly. Imagine you have several tasks that you would run in parallel at the bench, but the robot can only do like certain tasks in parallel and not others. So you need to think about that. You need to think about the displacement of the robot, like the parameters, as Christian said, the pipetting parameters, all these things. Like it's not intuitive for a scientist. Like they don't know what kind of parameters to pick for the pipette. So even if you had a no-code interface, you still have to tackle all these things. And so, of course, we get rid of the code, but we also get rid of all this lab automation knowledge that is required. So it's like a complete change in paradigm in terms of like ease of use.
Lab automation knowledge is a really important thing. Like, by chance, do you know how to program?
I'm no, very basic understanding. Well, I had to do for my neuroscience research. I had to do is learn the software language Xantex for this one device, but it was just my few weeks of my summer was just trying to understand that language. So I have that's like a painful area for me. But go ahead, please explain
for listeners who may be really into programming, I would challenge them if they want to understand this problem to go get a friend and give that friend a jar of peanut butter, a jar of jelly, and a few pieces of bread, and then try to write as detailed as possible an explanation of how to make a peanut butter and jelly sandwich. You can even try like formalizing this into a script or something, or writing really detailed pseudocode. And no matter how hard you try, despite you having programming knowledge, once you hand that sheet of paper over to your friend, they'll rip apart the instructions. You say, put jelly on the bread, they're going to put 1 gram because you didn't specify. There are all these things that we forget to specify. So the real barrier to entry to using these tools isn't even just programming knowledge. It's being able to specify so exactly within the constraints of lab automation. It's lab automation knowledge. It's really, really important, as Théo says, to automate to improve ease of use.
So it's essentially so much of the obvious stuff to us, like the implicit obvious stuff to us, is very hard to make it like understandable in programming language. And you guys are really getting rid of that. It's very, very cool. I'm sure, I think if I read it correctly, it's already being deployed in a research lab, your technology. Okay. Could you guys talk a bit more about, the real life use of Tetsuwan's technology right now?
Yeah, absolutely. It's been deployed in a lab called La Jolla Lab down in San Diego. It's a tiny startup. They have 3 people in the wet lab, pipetting all day. What they do is super exciting. They work on drug discovery for rare disease. So it's type of gene therapy. They use antisense oligonucleotides. It's a very long and fancy word, but basically, like Christian explained to me how it works, because I don't know biology, but I'm good at like explaining like at the like very high level. When you have a genetic disease, some of your DNA is bad, right? This DNA is translating to RNA, and this is translating to proteins, and those proteins have like bad effect on your cells, and you get sick because of these proteins. So ASOs are a little like molecule of DNA or RNA that comes into your cell and blocks the RNA so that it doesn't translate into protein that have harmful effects. Did I get it right?
Yeah.
Perfect. And so exactly, it comes and like blocks like the disease basically. And in order to get into the cell, they need to have like a key to bind to the receptor. It's called a conjugate. And there are very few conjugate discovered. There's only one for the liver.
Galnic was approved by the FDA a few years ago, and it's only FDA approved conjugate now.
Yeah, and they have like we have many types of cells, right? So we need to find conjugates for all like type of cells. So there we're running a conjugate program, and part of this is running a lot of qPCR experiments, like quantification experiments, and not a single qPCR is identical to the other into like parameters or like set of primers that they're putting on their samples. Like, and it's always different. They've tried automation, but it didn't work. They didn't hire a lab automation engineer, and therefore they couldn't like interface the scientist and the robot, and the program failed. So yeah, we deployed our system down in San Diego for them to be able to interface seamlessly with the robot and run their qPCR in a much more reproducible way.
Really, really cool. This technology has a potential to really, again, as I mentioned earlier, like fundamentally change the way we do science, right? One thing though, I've noticed from my own kind of research experience, scientists, healthcare professionals, and some other fields, like they tend to be resistant to change, especially when it comes to the way of doing things, to their methodology, right? Are you guys coming across some skepticism, or do you think like what's the I guess the current general attitude from the research and academia right now towards your technology?
Yeah, absolutely. Like you say, scientists, for good reason, are some of the biggest skeptics. There's a big graveyard of companies in our space. The pitch of every lab automation company since the inception of lab automation in 1990 has been that this is easy to use, flexible lab automation. These people have heard this pitch over and over and over the past few decades. So usually when we go to talk to them, they ask, what's different? What's changed? We just had this sales rep from a company 5 years ago tell us the same exact thing. So not only are scientists naturally skeptical, but there's a lot of failed attempts to try to do what we want to do now. In addition to that, there's a lot of skepticism still around lab automation itself, and a lot of miseducation, unfortunately. A lot of scientists perceive these tools to be black boxes. You're putting your super valuable cell line into this box that's going to use this iron arm on a gantry to come over and mess up your samples. You don't want to do that, right? Like, you don't know what's going to happen. You don't know if it's going to aspirate the samples wrong or something, or drop a tip. Like, it's seen as a black box that you throw really valuable things into, and you might not know what comes out. And this black box stigma is only really worsened when we talk about further removing the human translator from the picture. So we have to think of really, really smart ways to engineer transparency into this system that scientists may perceive as a black box. Think about how we can abstract 1,400 transfers to them so that they can catch and make sure that every transfer is correct, or how we can engineer closed loop systems to make sure that the robot doesn't crash and that the scientists can place trust in the system. So it's adoption is one of the biggest things that we think about.
Very fair. And the process of from the beginning, building the technology, and like obviously this is a deep tech technology which requires significant funding, and I believe you guys raised decent amount of funding, 2.7 million last time I checked, from ventures like 2048, carbon, silicon, and everywhere we see. What obviously VCs like to fund this cool technologies, but what did it take you to convince those investors to fund such a sci-fi like technology, right? Like what was the process like for you guys?
Yeah, unfortunately, when and if Théo and I walk into a room and we say that we think that scientific discovery itself is solvable and that it will be solved within our lifetimes, not much of an action plan there. Big vision, but not much of an action plan. So we have to piece together what we're doing right now in the short term and our long-term vision. That was the big challenge of getting venture support on this project. What we're doing in the short term does not seem to have obvious implications for automating scientific discovery itself. Very, very key to the problem of granting models physical agency. Models can't control these lab automation platforms, similar to how humans without automation engineering experience can control these platforms without this translator. So by automating this translation, we are working towards granting models physical agency. So being able to tie, hey, we're making automation engineer tools right now, but this is going towards the grand vision of granting physical agency to models to automate the process of scientific discovery. Bridging that gap and explaining to investors was, I think, what really made the difference.
Gotcha. This is really cool. And, do you, I guess it's kind of like a follow-up question I'm personally curious about, because different VCs invest in different kind of technologies, and you kind of have to find your, they say product market fit, you also kind of have to find your VC fit, right? Do you guys, when you guys pitch the idea, do you guys try to shape your pitch for the VC you're pitching to, or is it like a core kind of like a simple way of delivering just the idea to every VC you guys come across?
Yeah, I think there's a pretty simple way. It usually boils down to about 3 points. The first is that we believe that autonomous scientific discovery will be the most important field of our time. It will change the way that the world operates and science operates. The second is that automating the automation engineer is key to achieving that grand goal. And the third is we think we can make money automating the automation engineer until autonomous scientific discovery takes off, this much bigger field, right? Those are the 3 main points that we would explain to any investor. But the level of abstraction which we explain these points at varies based on the investor's background.
Gotcha. I actually forgot to ask one question. I just saw it on my list, which was about the point you made about reproducibility, which is a massive issue. I've seen it. I've seen it even in my own research experience, when you have to read some scientist methods and you see it doesn't really fit the way you're doing it, and then you have to switch it up a bit. And I think when I first moved to London to do my master's here, one of the first startups I interviewed for my podcast was Briefly Bio, which they're tackling this reproducibility issue in science and biology, particularly with AI. And I'm curious about both of your thoughts on that. It's a massive issue. It's a crisis, right? And it really adds to the complexity of the science, of the way we do science. I guess I'm curious about how you guys are taking that into consideration, and how you guys are addressing it when you're building the Tetsuwan, essentially.
Yeah, absolutely. You're 100% right. It's a huge problem. Reproducibility is crazy. You've seen it on your personal experience, and we've talked to a lot of people during our customer discovery phase. One of them was a former director of research at Pfizer. And he told us, like reproducibility is like really important for us. We want to reproduce like research that we've read in the literature because we're trying to build upon that, and it's like very interesting for our own like internal R&D. But my team of scientists, when they like take those papers and try to reproduce it, 70% of the papers they try to reproduce, they fail to reproduce. 70, which is insane. You hear about like reproducibility crisis in like other types of science, like human or social science, but when you 70% is insane. And this like comes down to like several problems. There is the ambiguities, and it's always like ambiguity problem in the protocol. You have the natural language ambiguities, right? Like when the protocol is like written in natural language, there's already some loss of information. The scientist might simply forget to write a step. That's happened when we deployed at La Jolla Lab. The scientists gave us a list of steps, and at one point we had a filter plate, and they asked us to put 70 mics. It was 100 mic capacity. They asked us to put 70 mic of a reagent and then 45 mics of another reagent. And we were like, wait, that doesn't add up. And they said, oh yeah, but you just need to apply vacuum and filter so that, it resets the volume between the 70 and the 45. It's like, oh yeah, like, of course it's obvious, like when you know it, when you have the context. But if you don't have the context, you cannot know that. So you can completely forget steps. Christian brought up the example of the peanut butter and jelly sandwich. That's like the perfect example of like how you can have ambiguities in the natural language. So having a system that is able to ask for feedback, get confirmation, and have like some context in order to clear all those natural language ambiguities is huge. And that's one part of the work that we're doing. Second part is the like actual actuation, like the actual way you perform the experiment. Now that you need to pipette 40 mics of that reagent into your well, how are you going to pipette it? This same senior director at Pfizer told us that sometimes they would have to send teams to the universities, to the faculty, to see the experiment performed because they were really interested in that one paper that they published. They couldn't reproduce it internally, so they sent a team of people to see. And yeah, that one PhD in that one university could do it just because of like the syndrome of like magic hands, like the way he was holding the pipette, the way he was pipetting, like maybe like he was pipetting very slowly, very carefully. You don't know. If you have a robot doing that, you can control every single parameter so that the pipetting is like absolutely perfectly reproducible. And the final thing is also how you evaluate your result. Like Christian's example of cooking earlier said, cook your pancake until it's like golden. Okay, like that applies to biology as well, right? The protocol is going to tell you mix until blurry. This isn't like something like Luis Villa, a guy from automation, like always like says, mix until blurry. How do you know what blurry is? That's just an example. So if you can smack computer vision here and have a way to quantify how blurry it is, like try to combine intuition with like more quantifiable methods. That's like key to solving these ambiguity problems. And like all these points, we're tackling them. And reproducibility really slows science. When you see the exponential improvements of models in AI, you see every week there's a new paper published. And when DeepSeek publishes like the R1 paper, everyone can just like download it on their laptop. They create a virtual environment, they clone the repo, and they can run the same exact algorithm, and then they can build upon that. And you just like get exponential growth based on that. Imagine you could do the same thing for biology. You have a repo with your experiments, and you just like clone this new paper that you just saw onto your robot. The robot runs the exact experiment and gets the exact same result. Like imagine how fast science would move. That's just like mind blowing to us.
No, you guys are solving so many things. I mean, 70% of the I didn't know about that. 70% is huge. I mean, it's costing probably billions and billions of dollars every year. That amount of reproducibility issue. Another thing is the way you guys are building the technology. It seems like it's very much like easily you can integrate it quite well seamlessly into existing workflows, which will make the life of the scientists quite quite easy as well. And another thing you mentioned, that doing science in that way, where like you can produce data and you can get the data and then build upon that, I think it's going to also help in like kind of move forward the science in general, right? Like to train AI, you need massive and massive sets of data. But if the data is obtained in different ways and different methodologies are used, you cannot really train, because you don't just need a massive data. You need data that's kind of like organized in it, so you can like draw conclusions from the data, comparisons, patterns, and whatnot. So genuinely, really, really exciting technology what you guys are developing. I guess one thing, you mentioned technology, you mentioned the first deployment. I'm curious about the future, like next 5 to 10 years. Hopefully, more exciting news gonna come soon from you guys. Do you guys have any certain milestones and plans ahead?
Yeah, of course. We are targeting a total of 5 deployments by the end of this year. We want to show that these systems are generalizable. We're not
Mhm.
building tools that do a single protocol or something like that. That this is a generalizable translation architecture that is useful across different labs and different settings for different experiments. So 5 of these systems running by end of year. And we also have plans and are currently working on an internal project to kind of make the same jump for lay people that we made with investors, of saying, okay, what does it mean to automate the automation engineer? Why is that important? Why do I care about that? The people that we want to hire, the new grads out of MIT or Caltech or Stanford or wherever, they don't run to lab automation. This is an old, small industry, right? So we have to be able to tie this problem that exists within an old, small industry into this much bigger new industry. Why is automating the automation engineer important? Can we showcase that in a way where it's vivid to anybody? Why it's consequential? What physical agency granted to models might look like for scientific discovery? We have to bridge this gap and be able to show it to the rest of the world.
Fair, very fair. Gentlemen, we are coming towards the end of our episode, and I have 2 more questions, I believe, to go. One of them is going to be more philosophical in nature. I'm curious. It's up to you to answer in any way you guys want. AI is already changing the way we live and the way we work. Some of them in a pretty good ways in science and healthcare, but they're potentially concerning ways too, right? And I was reading in your blog, I believe you mentioned the concept and idea of duplicator, which is really interesting, right? Like this genuinely super sci-fi. It's an AI scientist with PhD level capabilities. But that made me think about like the role of a scientist once it's something, the technology of that caliber is deployed, right? Like what will be the role of a scientist? Will scientists essentially be replaced and outpaced in innovation and finally be replaced? Or is it just going to be a tool to help scientists essentially always act as a tool to help scientists to augment their creativity and just like work more efficiently? What do you guys think?
If you look at our logo, it's these 2 triangles that are flipped on top of each other. The first triangle that looks like a normal pyramid, we made it that way because our frustration with research is that it's very top down. You have
Mm,
a PI or you have some senior staff scientists, and then you have more people below them. And then at the lowest level, you have a whole bunch of RAs. It's a pyramid, right? It takes a long time to climb to the top of the pyramid. And we have this pyramid structure because so much of our R&D, so much of our discovery relies on repetitive physical labor. We need these people at the bottom to conduct that repetitive physical labor, right? But what
mm,
happens when we automate that repetitive physical labor away? We think we turn the pyramid on its head and it forms another triangle where any scientist can act with the capacity of a PI in direct labor to explore their own ideas. That's what we're really, really excited about. Like you say, we read these pop sci magazines when we're
yeah, yeah.
kids. We watch Jurassic Park. We think about like wonderful, wonderful things that inspire us. When we go into research, we should have that sense of agency that we believe that we would have had as scientists as kids. And what our work is about granting more agency to these human scientists to focus on the intellectual part of discovery and turning the physical labor part on its head. So then you get this little hourglass shape.
That's fantastic. I'm really really excited to see. I'm gonna follow you guys to see all the news that come out of your startup. I guess there are a lot of people that watch this episode. I've interviewed people building in biotech, consumer tech, fintech, etc. And a lot of people who watch these episodes are like tend to be students in university, doing PhD, masters, undergrads who want to do their startups, who want to find their company, and I'm curious, what advice would you guys have to young founders, especially deep tech founders, which is a very complicated area to build in, very capital intensive too. What advice would you guys have for them? And I guess some of them might be watching you and be inspired by your story too. So stage is yours.
Do you want to go?
Yeah, we'll both give our.
Yeah, we both have one answer.
Yeah, I try to smack this into everybody's heads whenever I get the opportunity. Probably a broken record by now, but I tell anybody who has an interest in entrepreneurship that entrepreneurship is not what you believe it to be. Entrepreneurship is about journalism. There'd be no good mechanics if there were no physicists. You have to understand physics to be able to build a good mechanical system. You have to understand the way that things interact to be able to harness this, right? The same is true for our work. We need to be able to rigorously understand human behavior. Why do people do the things that they do? Why haven't incumbents solved this issue? How will people react to this change? Why do people make the purchasing decisions that they make? All of these decisions, they are captured by incentive structures that people exist within. Incentive structures are like physics for people. If you have a really, really rigorous understanding of why people do the things that they do, you can affect that behavior. And that's what we're supposed to do. As entrepreneurs, we create technology to affect behavior. Ideally, this makes the market more efficient, right? But you can't affect behavior without understanding behavior in the first point. So I would really, really push as heavily as possible that even if you're in deep tech, even if you've bought into the social network image of what Silicon Valley is, this misunderstood genius in a hoodie that doesn't talk to anybody. That's all wrong. Like, this is all about journalism. This is all about incentive structures. This is all about people and the way that they behave. So look at it from a journalistic perspective and also choose an important problem. I think we both buy into the belief that this is the most important generation to ever live in terms of the technical impact that we will have. If we mess up the set of technical problems that we've been given, it may be game over. So don't do something stupid. Don't work on some grift Web3 thing. The time is now to do something meaningful. So go find a meaningful problem.
This is, Theo, I'm curious about your perspective too, but I just want to say Christian is one of the most genuine and unique perspectives I've heard in a while. Usually you get very much like the standard answers, but I really appreciate your perspective, and genuinely very inspiring to see two young guys, pursuing this very genuinely complex and complicated idea and actually want to bring it into market. So very inspiring story from both of you. But I'm curious about your perspective too, Sarah.
Yeah, absolutely. I agree 100% with Christian. And for me, the perfect transition. He said, don't be like this lonely guy in your hoodie, not talking to anyone, but talk to people and do journalism. Talking to people is not only good for journalism and really understanding the problem, but building a company relies on knowing the right people. You cannot build a company on your own. Tall trees grow in forests, and you need to know people. And actually, university is a perfect place where it's a perfect ecosystem with very highly driven, highly ambitious people, high impact people. Don't stay in your room. Where I'm from, I mean, most universities, students are very focused on passing exams and studying for assignments and everything, but don't miss out on the opportunity of talking to so many people at university. Your professors can maybe later become your advisors. Alumni of your university might have built companies and can become business partners. Students that are in your class might become your co-founder or your first employees, or even talent hunter for you. You really need to know people. So get outside of your room and go talk to people.
One, if I could ask one more follow-up question, when it comes to, you guys seem like a perfect combination duo. What advice would you guys have for finding your right co-founders for yourself?
I think you got to balance each other out. I think if I didn't have Théo or somebody exactly like Théo, none of this would work.
Gotcha.
You can't have two versions of yourself. And I think people tend to go in that direction because it results in less hardship, right?
Mm-hmm.
If you have somebody who always agrees with everything that you say and everything is just a duplicate of what you're doing, it's going to seem great, but it's like eating really fatty foods. It's not good for you. It's not healthy. It's not sustainable. So Théo is incredible, an incredible technical leader. I think one of the most organized and thoughtful people I know. But me, like, I can't organize things. Like Théo put together a Notion, but the state of our documentation before that was, not good. So I think you need to balance who you are as co-founders and reach the right level.
Yeah, absolutely. I mean, like Christian's extremely driven, like, it's like so ambitious. And like, I'm always the one who's like kind of like dragging my feet. I'm very skeptical, like Christian always tell me like, oh, like we have this idea, but you always have a but. And I'm like, yeah, like that's the way I think. Like I try to find all the issues that would come up and then like we solve them and then I give the green light,
Yeah.
So it's like, if it was just you, like we wouldn't do anything. I was like, yeah, I know, but like we got to make sure everything happens. So, it's like really like complementary. It's like,
yeah, if it was me, we'd probably try to be building a time machine in the back there.
No, gentlemen, this was a wonderful conversation. I genuinely appreciate your drive and what you're trying to do, not just for the science, but in a way for humanity too. And really, really inspiring story how you guys really came together, started this company. Looking forward to a lot of good news from you. Hopefully, we can stay in touch. But that's pretty much it. This is the end of our episode. Thank you again so much for your time.
Thank you, Javid. Yeah.
Thank you.